Hello BIOL1009, welcome to week 6. We're continuing on our study of DNA, how DNA generates the material that makes up life, i.e. Proteins, enzymes. And so today, this week, we're going to be learning about the first step in that process, i.e. taking the information that's encoded in DNA and translating it into an actual polypeptide or a protein. Polypeptide being a string of amino acids and a protein being a very long polypeptide that is folded into a shape that actually serves some sort of function. It drives some chemical reaction. It has a structural purpose like a skin cell. Chapter 11 is focused on that process of transcription, so going from DNA into RNA and then into translation, going from that RNA molecule into an actual protein molecule. So that's where we're going to learn about that process of transcription and translation. We call this gene expression. How does a gene that's encoded in DNA get expressed into a functioning protein or enzyme? That's chapter 11, and then what we're going to be moving on in chapter 12 is how that process can go awry due to either mutations, how mutations can disrupt that, and potentially how mutations can lead to the development of cancer. So we're going to be connecting this process of gene expression and looking at exceptions to that, where those processes go wrong and how mutations underlie that disruption. Keep in mind, not all mutations are bad. Mutations just change the structure of a DNA sequence, which can have subsequent consequences in terms of the gene that is, the protein that is expressed. Not all of those changes can be bad. Mutations are really the ultimate source of variation amongst populations, and some mutations can be beneficial. A lot of times, though, they're not. And so that's what we're going to be learning about in chapter 12. Standard issue week, you have your Learn Smart material, chapters 11 and 12. You have your online quiz. You have your homework, which you need to get done with your group. For the lab this week, I'm going to be reviewing your experimental designs for lab 3, so the animal digestion lab. I'll be looking at what you guys proposed, giving you suggestions for how you might do it a little more effectively, making sure everything is good to go before you actually carry out the experiment. And then in lieu of you doing anything with lab 3, you're going to do lab 4, which is an online activity, an online module focused on photosynthesis and respiration. So you should have some familiarity with those subjects because you've already read 2 chapters on that material. And this is really going to walk you through how they're related to each other and how they relate to sort of human energy consumption, energy transfer from one phototrophic level to the next, and kind of give you a little more integrated understanding of those 2 concepts, because they're going to come back into play when we get into ecology, because ecology is the study of how energy moves through communities. So we're carrying this information forward. All right. Good luck. If you have any questions, obviously reach out to me at the TA Lindsay. We're happy to help you with this material. It's a little dense, so do not hesitate to reach out to us. There's a lot of information coming at you in these 2 chapters. So please, if anything at all doesn't make sense, let us know. We're happy to help. Otherwise, have a good week, and I'll talk to you later.